Dr. Francis Keach.
Good morning. As president of the Education and Research Foundation for Nuclear Medicine and Molecular Imaging, ERF, I am most proud of the fact that we have funded virtually all of this SNMMI annual grants and awards and scholarships for the past 47 years. We put your gifts to work funding the most rigorously vetted physicians, scientists, and technologists in the pursuit of advanced academic studies and breakthrough discoveries in the field. One of the hallmark programs of the ERF is the prestigious prize named in honor of the late physicist Benedict Cassen, whose invention, the rectilinear scanner, first brought the ability to image patients to nuclear medicine. His widow, Mary Wiley Cassen, made a bequest to the foundation to establish this major award. Benedict Cassen was indeed a pioneer in our field, and it is fitting that the Cassen Prize bears his name. The Cassens were leaders by example, both in science and philanthropy. And now, I'd like to introduce Dr. Michael Phelps, the Norton Simon Professor, Chair of the Department of Molecular and Medical Pharmacology, Director of the Crump Institute of Molecular Imaging at the David Geffen School of Medicine at UCLA, who will share with us a little bit about this year's Cassen Prize winner.
It's my privilege to say a few words about Sam Gambhir. So, here's his family. His family immigrated from India to America. You see his mother, who's a school teacher, his father, a very successful businessman. In the bottom right-hand corner, you see little 5-year-old Sam. Oh. So, in 1994, after Sam had completed his MD and his PhD in biomathematics, his internship and residency in nuclear medicine, and became an assistant professor in my department, he came to talk to me. And he said that he was not sure that people would ever be interested in his work that brought together physics, biomath, and nuclear medicine. I said, Sam, trust me. You have the ability, the imagination, the relentless drive to not only create interest in our profession, but also to build a new future for it, and you will be fine. Now, this is a picture, a photo of Sam when he was becoming a boxer. Now, Sam and I share many things in our life, but boxing is not one of them. Now, Sam entered Arizona State University at 15 years of age, where he got a BS degree in physics. He entered the MD PhD program at UCLA when he was 19, where he got his MD degree and a PhD in biomath. He quickly went from an assistant professor to a full professor, became a vice chair of our department, and director of the Crump Institute. And then Stanford recruited Sam, and you see the 6 titles, administrative and scholarship above all else responsibilities of Sam at Stanford. Now, a few academic facts about Sam. His total funding from grants and donor money and foundations is over $250 million. He has 635 peer-reviewed papers. 40 of those appeared as covers to illustrate the importance of the publications. 3 books and 1 ebook. Amazingly, he has the single largest funded program at Stanford University, and it's Stanford University, one of the finest universities in the world. He's educated over 250 students that have gone out all around the world to build their careers. Now, I said that Sam and I share many things, and we share many good things, but we also share tragedy. And Sam and Arun, his son, Millen, wonderful, beautiful son, died last year from a glioblastoma. Now, in my family, my 2 younger brothers, Butch and Jimmy, burnt to death when our house burned down. And my mom was burnt over 50% of her body. So we share great things, we share tragedies. Now, the Irish have a saying that you should never regret what never was, but rather cherish the time you had together. Now, today, if somebody asked me who Sam Gambhir is professionally, I'd say whatever the hell he wants to be. So I introduce my student, my colleague, and my dear friend, Sam Gambhir.
Thank you so much, Mike, for those very endearing words. Thank you all for giving me this honor and privilege of being in front of you today. This will be somewhat of an emotional talk in that I will tell you many things that happened along this journey in the last few decades that involved some fundamental biology that was going on in my son that would lead to his death. But I hope you will understand and allow me the privilege of being able to explain certain things to you. Some disclosures. So I give this talk in honor of my son, who died 3 years ago of a glioblastoma, a brain tumor. Milan, just like many of your children, was a wonderful human being, had so much in front of him, and all my wife Aruna and I can do is honor his memory by trying to do many of the things he believed in. He had attended this meeting as a young child, and I'm sure would be in some way or another involved in this field. And I will tell you a few things about some of the things he was thinking about towards the end of this talk that I think will impact our field and ways that I'm trying to move our field forward as well. I'll start off with some basic cell biology and reporter gene technology to get everyone on the same page. We'll move quickly to the imaging of gene therapy as one application in humans that I think will go on to play major roles in human therapeutics. Then we'll move on to cell transplantation and trafficking and new technologies we've been developing in gene amplification and what's called split reporter gene technology. And finally, as I shift gears a little bit towards the end, I'll give you some thoughts on how we're taking healthcare at Stanford in a new area that's not precision medicine, but precision health. And then I'll end with concluding remarks. So we're all used to seeing the phenotype of each other, of animals we work with, of research we do on a daily basis. This phenotype, of course, is governed by trillions of cells that underlie what we see with our human eyes, and over 3 billion DNA base pairs and 30,000 genes, leading to about 100,000 different proteins. When I started to understand cell molecular biology to a greater depth at UCLA, working with Dr. Harvey Hirschman, another mentor, I started to appreciate the complexity of the underlying biology that was governing the phenotype. At that time, I didn't fully appreciate one particular gene, the p53 gene, but it would grow to be one of the most important genes in our family. Milan appeared just as any child would and as any teenager would, but in each and every one of his cells was a mutation of the p53 gene that would predispose every cell in his body to cancer. In fact, he would have a 85% lifetime risk of cancer, and had he been a female, he would have had a 100% lifetime risk of cancer. This is known as Lee-Fermani syndrome, in which you have a mutation in the p53 gene. So what's important to understand is in all of us, of course, there are likely mutations of many different genes, only some of which we understand at the current time. And of course, as evolution would have it, and as natural selection would have it, those genes that are more significant, mutations that are more significant, of course, lead to early death. Now, neurons, hepatocytes, other cells are, of course, quite different from each other in their phenotype, but they have the same exact genes. And to cell molecular biologists and developmental biologists in particular, this has been a fascinating story of understanding how, in fact, cells can have the same genes, but yet go on to have quite different functions. At the core of this is that every gene has a promoter driving the expression of that gene, shown here as a darker red color. That promoter leads to transcription of the gene, and that transcription leads to translation to protein. Of course, what has led to a gene being on in one cell and a gene being off in another cell is in fact governed by many complex interactions, including proteins themselves. And as you know, these proteins have many features and functions within cells. Some form the structure of the cell, giving it its three-dimensional shape. Some go on to become receptors that we target in nuclear medicine and molecular imaging. Some go on to become transporters that we take advantage of to get our imaging agents and our therapeutic agents into cells. Some are enzymes. Some are involved in signal transduction, relaying things from the outside of the cell to the inside of the cell. And some, of course, are themselves involved in the process of regulating gene transcription and mRNA translation. These protein functions are enormously complex and yet open up many possibilities for what will be the future of our field. We've been very good at targeting elements of these because we've borrowed tools from many fields to go after these when, in fact, we think they're important biomarkers of various diseases. Intrinsic to all of this is that much of ways that we can think about gene transcription is like a light dial switch. The promoter, enhancer elements, not always just in front of a gene, not always just in front of exons and intervening sequences or introns, are driving the expression of that gene so that one can produce more and more message, and sometimes that more and more message leads to more and more protein. If we can borrow these switches from one gene and put them in front of another gene, we have the ability to rewire the entire network of a cell. And in fact, cell molecular biologists constantly clone individual regulatory elements like the enhancer and promoter in front of one gene and use those to play tricks, if you will, within the cell. This became clear to us as a field when, in the late 90s, we started to look to see how we might bring the field of cell molecular biology together with the field of biomedical imaging and nuclear medicine. Our traditional approaches are always based on going after a known target. In this work that I show as an example, which is a recent collaboration between Stanford and Harbin in China, where we're doing more and more work in general, you see that we've gone after the epidermal growth factor receptor, or EGFR, and specifically found ways to image the mutant EGFR. The drug in this case that's modified to allow for better biodistribution is labeled with fluorine 18, and the CT images shown on your left, the FDG images in the middle, and the new tracer that targets the mutant EGFR on the right are used to, in fact, do a sampling of this known mutation within lung cancer. What you see in the top row is that the new tracer, 18F-MPG, shows signal in the lung at the lung tumor site. Similarly, the middle row shows signal there as well, indicating that a mutation in EGFR has been imaged. And in the bottom, you see no signal in the 18F-MPG lung images because that individual carries the wild-type EGFR and not the mutant EGFR. This lets us, of course, do non-invasively sampling of the entire body, in this case, looking for a specific mutation in a gene, in this case, a very important gene in growth regulation. I show this as an example only because most of our field is very focused on having to deal with what nature provides already. we don't change the biomarkers in general. We don't go in and force cells to make a mutant EGFR. We don't force cells to make PSMA. But what we will see in the reporter gene work is that we're able to flip the paradigm around and force cells to do things they wouldn't otherwise do, and thereby open up entirely new ways of tracking processes within the body. That's really what the reporter gene is intended to do. It's intended to report on some other event within a cell. If we can deliver a reporter gene into a cell, in this case driven by a red switch of choice, the dark red switch, we can again force these cells to make a protein that it may not have otherwise made. Or it may be a protein that it used to make a long time ago, but has shut down the expression of that protein as the cell has matured. The key is this is a gene that is now being turned on and transcribed and translated, and now that protein that's being made can be very useful to us. We can, in fact, force that cell to secrete that protein so that the protein can end up in the blood or in the urine, and thereby we know somewhere in the body a gene has come on that has made that protein. We can, in fact, use that protein to image the location of the cells that are producing that reporter gene. In this case, a reporter probe or a tracer is now targeting that protein either as a receptor-based interaction or as an enzymatic-based interaction. But the key is that that protein is allowing a signal to occur that otherwise wouldn't occur. Because the spheres that you see are tracer, if it went to a cell that didn't express the protein, would not accumulate in that cell, thereby letting us specifically detect reporter gene expression. That simple concept, which occurred to several groups around the world at around the same time, led to a lot of the things that I'll show you. And that concept let us rethink how nuclear medicine could evolve in ways that are now paying off as cell therapies come to full maturation, including in immunotherapy. This process can be very interesting if you take advantage of it in unique ways. Shown on the left is a blue gene that is driven by a red switch, a red promoter. And what you see is a blue protein being made. But imagine that I don't have a tracer for the blue protein. Instead of building a tracer for the blue protein, I can instead deliver a reporter gene into that same cell, shown as the red reporter gene. And now I can make red message and red protein, but I have an imaging agent for the red protein. That imaging agent for the red protein lets me indirectly infer what's going on with the blue protein. This was very intriguing to me because it freed me up from having to develop ways to do chemistry and radiochemistry. It said we could always go back and study any process in a cell by simply engineering that cell to make a reporter protein for which we already had an imaging agent. And then by simply changing the switch out in front of the gene over and over and over, I could study many different things within that cell without having a new tracer for that cell each and every time. This then led to understanding how to make this work in general, and one such approach was through the HSV1-TK suicide gene. This suicide gene has been developed in the 80s as a potential way to kill cells. The idea being that if you had the HSV1-TK gene being expressed in a cell, and now you gave a prodrug like pencyclovir shown here, that prodrug would be phosphorylated by the HSV1-TK enzyme, and that phosphorylated prodrug would be trapped in the cell, and when given in pharmacological quantities, that prodrug being phosphorylated would interfere with DNA replication. That would eventually lead to cell death. This is the reason that when you get a herpes virus infection, that if you treat that viral infection with creams that include creams that are acycloguanosines, you're able to, in fact, kill cells that affect and are affected by the herpes virus and spare cells that are not affected by the herpes virus. This same concept can then be borrowed to convert this to an imaging reporter. HSV1-TK can be imaged by having, for example, an acycloguanosine labeled with fluorine 18, and now this fluorohydroxybutylguanine will go into a cell, will also be phosphorylated if and only if the gene is present in that cell and is being expressed, a gene that normally would not be in your cells, and thereby, because it's trapped in the cells after phosphorylation, it would lead to signal letting you know that gene expression is occurring in that cell. Of course, cells that don't have this gene would still have the probe enter the cell, but your normal thymidine kinase does not significantly phosphorylate this agent, thereby letting the agent go right back out since it's not phosphorylated. So HSV1-TK would become both a suicide gene and a reporter gene. This would become critical as we convince the FDA to let us move these applications into humans, as you'll soon see. Many people in the field would work on multiple reporter genes that would be at the root of being able to have a gene that's not normally expressed in your cells and a corresponding imaging agent that would interrogate the expression of that gene. This would occur with PET and SPECT-based reporters. It would also occur with optical imaging, including GFP, the luciferases, and then even secreted reporters, reporters that will end up in the blood that we'll take advantage of, that you'll see at the end of the talk, to detect cancer early. All of these continue to grow with more and more findings of new ways of taking advantage of this reporter gene concept. So how did we move this into imaging of gene therapy? One of the first things that we did was we looked at adenoviral infection within an animal model. If you have an adenovirus that carries this reporter gene and that adenovirus infects the cell, now that cell will be able to trap, in this case, an acycloguanosine, whether it be fluorine 18 ganciclovir, as we originally used, or now fluorine 18 penciclovir. The mouse on the left bottom is a mouse that's been injected with an adenovirus, and that adenovirus has infected the liver, and then 48 hours later, we've given the tracer that should only accumulate in cells that are expressing this gene. And in fact, you don't see any significant liver signal. All you see is the significant bladder signal as the probe is excreted. But the mouse on the bottom right shows you a lot of liver signal because the gene has been successfully delivered to the liver and is in fact being expressed in hepatocytes and is thereby trapping the PET tracer through phosphorylation. In fact, in those days, the micropet, one of the real reasons we were exploring it is that when I started research, we would always do dogs, sheep, other large animal models. But we were starting to make a shift towards small animal models, and this was one of the key applications, was being able to image transgenic mice, mice that we had delivered genes into with a dedicated small animal PET system. And that's what would give rise, through Simon Cherry and other people's efforts, in the micropet technology. This just shows you that as you look at the tracer moving around the animal, very quickly over 30 minutes it's excreted, and in fact then over the last 20 minutes or so starts to build up in the liver, which may or may not project well, but that liver signal is what you see in the static image on the bottom left mouse. This pharmacokinetic optimization actually took us many years. Although it seems straightforward here, we would undergo a lot of failures before it would work. And others also working on this, like Dr. Ron Blasberg and Yuri Gelovani, were trying different approaches with different tracers that I'll come back to towards the end. But in 1998, this then would come together and was on the cover of J&M as we first looked at moving these approaches towards human applications. We translated this tracer. Shahryar Yagoubi in my lab helped move this tracer to humans. This is the first human that was ever imaged with this tracer. This is a normal volunteer. What you're seeing is 60 minutes after injection of the tracer, clearance of the tracer into the gallbladder, the kidneys, and the urinary bladder, and very low background signal everywhere else, including the brain, where the tracer does not enter because it does not cross the BBB in this case. This particular image convinced us that there was good potential for this, as we now would start to actually do gene therapy in humans. Working with Dr. Peñuelas and colleagues in Spain, in Pamplona, Spain, we did the first imaging of adenoviral-mediated human gene therapy. In this case, adenovirus, about 1 trillion particles, 10 to the 12th particles, were injected into hepatocellular cancer in the liver in the hopes of destroying that cancer by using the suicide gene. Because that suicide gene is also able to be imaged, it let us inject into these same patients tracer that would let us image where the virus had gone, where it had successfully transfected cells leading to gene expression. You can see signal in that top row of a very hot area, and that's where the virus has been delivered near the dome of the liver. And in the fusion image in the bottom, you can see the CT and PET combined. More importantly, this showed us where the virus was not going. It showed us that it was not spreading to other parts of the body. Also, although I don't have time to get into it, it would teach us a lot of reasons gene therapy was failing, including that when you re-inject the virus, that there would not be gene expression because of an immune response preventing that gene expression from occurring. This would open up then all kinds of opportunities for imaging gene therapy in humans, which I believe will see a renaissance as CRISPR-Cas9 and human gene editing now starts to take off. And nuclear medicine will be right there with it to be able to actually image gene expression in humans. Dr. Contag and colleagues were studying the firefly luciferase gene at Stanford when I was still at UCLA. And we were intrigued by the fact that he was looking at very similar things to what we were doing, but with optical imaging. My lab then started to explore another optical reporter known as the Ranilla luciferase, taken from a sea organism, and selenterrazine. In this case, again, a probe is injected into an animal, but it's not radioactive. That probe produces light only when it combines with its target enzyme, which is encoded by a gene that you've delivered, and again, into the liver of this animal. This opened up all kinds of optical imaging approaches that would then lead to a huge growth in biological models that are now commonly used for optical imaging of gene expression. We would also work a lot with the late Roger Chen, who had been developing multiple fluorescent reporters to develop fusion reporter genes. Here we would fuse together luciferases, fluorescent genes, and PET reporter genes so that one combined protein is capable of fluorescing, interacting with substrates to produce light through bioluminescence, and capable of phosphorylating a PET tracer. It's actually one large protein, and the optimization of this actually took many years. This would let you go from cells to small animal models all the way to humans with a single reporter construct. We would then start to look at this with regards to a lot of cell transplantation and trafficking. As you know, nuclear medicine for decades, through the hard efforts of many in this room, has been able to do white blood cell imaging through labeling of cells with, for example, indium-oxine. Similarly, you could label cells with MR contrast agents or optical agents, inject them into subjects, and then track where those cells are going. This is powerful, but it doesn't let you follow the cells forever. It only lets you follow the cells for a limited period of time, and as cells divide, the label gets diluted. You can also, of course, go after cells by directly imaging targets on those cells that are unique to those cells. For example, if you have an immune cell with a cell surface target that's unique, you can now inject a radioactive tracer and track that cell and only that cell. The problem with this approach is you've got to build a new tracer for each and every new target that you want to go after. Reporter genes give you this interesting opportunity where if you're willing to tag your cells with a reporter gene, now you can follow those cells repeatedly for the rest of their lifetime, because even as they divide, each divided daughter cell will carry the reporter gene. and the imaging agent that you will use will then go on to let you identify only those cells in which the gene is being expressed. We would use this very early on to start to study the imaging of leukemia in vivo. Dr. Owen Whitty, working with individuals in my lab, would start to look at the behavior of leukemia for the first time and how leukemia cells in the bone marrow would come to the lungs as they would evolve in their maturation. That would then lead to a greater understanding of the biology of how cells trafficked in a living animal, in this case, before bioluminescence imaging actually started to take off. These approaches would be applied to multiple other areas. For example, Joe Wu, now the head of the Cardiovascular Institute at Stanford, would apply all these approaches to looking at cardiac imaging of stem cell transplantation. By labeling the cells, we would be able to determine through reporter gene expression where those cells are the cells leaking out of the heart, where else they're going, and be able to fundamentally improve what are, in this case, stem cell therapies for myocardial repair. We would also be able to take these same approaches, and if the cells got out of control, because the cells are marked with this reporter gene that can also be a suicide gene, if the cells were starting to grow uncontrollably, for example, in this case, in cardiac implantation that is spread throughout the mouse, we could give the prodrug, ganciclovir or penciclovir, and wipe out all those cells. This would be good evidence for the FDA that as we move to humans, that not only will we be able to image reporter genes, but we would help those cells that are misbehaving, if needed, to be wiped out through a drug that would only target those cells. We would also apply these approaches in multiple other areas. In the pancreas, looking at pancreatic islet transplantation, which is often done in the renal capsule, using switches that are not just always on, but switches like the rat insulin promoter, and those, in this case, driving a PET reporter gene. Also, although we won't have time to discuss it, all of this became very quantitative. We were not able to just get the images in terms of percent ID per gram, but actual levels of target cells that are expressing that reporter. This would eventually lead to one of the most difficult trials we've ever done in humans. This trial took a decade to do. It involved the City of Hope with Mike Jensen, who's now up in Seattle, UCLA, Stanford, and it's something that I feel very proud of because we stuck with it for over a decade to bring this into humans. Oddly enough, this same trial would be the same disease that my son would die from. When we started this trial, I had no idea, of course, that Milan would himself come down with glioblastoma. In fact, as he started to get different immunotherapies, I knew those immunotherapies would likely fail. And I knew why they would fail because of the studies we had done in this work. This work would now be referred to as CAR T-cell therapy and CAR T-cell imaging. What it really was at the time was taking mononuclear cells from a patient that has already been diagnosed with glioblastoma, changing those cells, genetically re-engineering them by putting in the reporter gene, shown here as hygromycin TK, also putting in another gene known as the IL-13 zetokine that would help these cells do a better job tackling the IL-13 receptor in glioblastoma cells. You can see by illustration the cells changing from red to green here. These green cells would then be expanded ex vivo and then infused back into the brain tumor. This was a very difficult trial because the patients are extremely ill. The FDA had never seen anything quite like this, in which we had to get approval to not only use a new tracer, a new reporter gene, a new therapeutic approach. All of this had to come together, but set the way for what are now being able to use CAR T-cell imaging approaches with reporter genes. This work would show us what was happening, interestingly enough, in a human that was getting T-cells and why those T-cells were potentially failing. We would be able to get images before cell implantation and post-cell implantation. The cells could be infused right into the brain or intravenously. In this image, they're being infused right into the brain. And what you're seeing is a PET MR where these cytolytic T cells that are marked with reporter genes are coming in, and on the left, you're seeing a lack of significant PET signal. On the right, you're seeing a lot of PET signal because now these T cells have home to the brain tumor that carry the reporter gene. As I slice from superior to inferior, you will not see any PET signal because we removed it. As I come back up superiorly, I've superimposed the PET signal. So very low signal on the left because we haven't yet infused the cells. When we come to the right, what you'll see, we hold back on the PET signal, but as we come back up superiorly, I'll superimpose it. You'll see that we can clearly see the cells homing to, in this case, the GBM. The FDA initially only let us do this once. Then they let us do it twice, pre and post. And then initially it was for recurrent GBM, and then we moved to initial presentation of GBM. I always felt sorry for these patients because I knew we were putting them through a very challenging therapeutic that was not likely going to work. We knew that they were putting not only their lives right up on the line for us, but also letting us do extra procedures like the imaging. My own son, several years later, would undergo a lot of these same studies. But unfortunately, because GBM mutates so quickly, we're not able to keep up with it. We're not able to have these T cells adapt with the mutations. That's actually, I believe, the fundamental limitation in these therapies, is that the T cells are programmed to recognize one target, in this case, an IL-13 receptor. But that target is mutating so quickly, and you're applying a selection pressure that you just can't keep up with that rate of mutation. What you need are immunotherapies that co-evolve with the tumor's evolution itself. These cells would nicely be visible, and we would start to understand why they're failing. We'd also start to see that the cells can move to other areas of the brain and areas that we didn't even know GBM had spread to. The area on the left where the arrow is pointing is a known GBM. The area on the right was unknown to us. And that's because cells had migrated there and found that area of GBM and engaged with it. Of course, these images are only following where the cells are going, not whether they are actually activated. But that's where I see the beauty of these approaches eventually playing out as cell therapies will become routine. We will not only be able to track cells and where they go, but using the same concept I showed earlier, we'll be able to trigger reporter genes to come on if and only if the cell recognizes a target, or if and only if the cell's machinery has been activated to kill a tumor cell, let's say through the granzyme promoter. Promoters can be swapped out in front of the reporter gene so that you're not just imaging where cells are, but fundamentally imaging the machinery inside the cells as they start to do a given piece of work. We would also spend a lot of time in gene amplification and split reporter technologies. Just to show you here things that I think will pay off in the years to come, we develop multiple ways so that when a gene is weakly transcribed, so that not much messenger RNA is made, what can we do to make a strong promoter drive that gene? We would develop these systems called Gal4 VP16 fusions, in which you have a tissue-specific promoter driving an intermediate system, and that intermediate system then drives your gene of interest, like your PET reporter gene or your optical reporter gene. Shown here is a mouse in which you would never see gene expression on the left, and then when we use this amplification system, that you would see that gene being expressed on the right. We would also develop approaches to fundamentally hijack the splicing machinery of any cell. In this case, when you have a gene making a message making a protein, the way our field has typically come after message is by developing radioactive antisense oligonucleotides, or RASONs. What if you could instead send in a pre-trans-splicing molecule, a PTM? That molecule can interact with the message of any gene that you choose, and now a hybrid protein is made, and that hybrid protein can be imaged, letting you, in fact, interrogate any message within a cell. These approaches, which I think are still going to come back in a big way, will be fundamental to letting us do many things without developing new tracers for each and every gene of interest and each and every protein of interest. Several students in the lab would spend several years splitting genes. We would split genes so as to make two segments that are not capable of producing signal. The idea being that now these two split parts are quiet, but if brought close enough together, they would activate and produce signal. This would lead to the splitting of HSV1-TK, which Tariq Masood, a radiologist who came from Cambridge and spent 5 years in the lab, would spend his 5 years doing for his PhD. That split reporter would then be used to accomplish what's called complementation and reconstitution. In this way, we could monitor the machinery of any cell by looking at X and Y within a cell. And when X and Y come together, the split reporters would come together, and now the split reporters would either produce light or would be capable of phosphorylating a tracer and thereby trapping it. These approaches are fully ready, and I believe as human therapies catch up, will be deployed in humans as well. Many other things would also take place over a course of a decade, some that we're still presenting here. For example, Tom Haywood, a great chemist from University College London, has been presenting work at this meeting on reporter genes that involve the blood-brain barrier and crossing the blood-brain barrier. And immunogenicity reporters would be studied very nicely by Dr. Yuri Gelovani. A lot of these approaches would be very important to eventual use of these kinds of approaches. So in the last few minutes, let me show you where I believe things need to go and where we're taking things with all of this work. As in my own son's case, if he had been born a few decades from now, I believe that we would have much better tools for risk assessment at all stages of life. We would know through family history, genetic screening, even socioeconomic factors, exactly what diseases a given individual is at risk for, not just at the time of birth, but at the time of conception. These risk assessment profiles will let us monitor each and every person in a different way. Why should you be monitored the same way as your neighbor? Why should you be monitored in the same way as your spouse? You have different risks for diseases, and your monitoring should be different. There will be enormous growth in customized monitoring. We're already seeing it in the Bay Area. Huge amount of tools that are coming up the pipeline that will be in your house, that will be on your body, that will allow us to monitor you based on your risks. That will lead then to the data analytics that will bring all this together along with the interventions that we're all used to currently. But instead of focusing on the tail end of the problem, the field will start to shift towards the front end of the problem. In almost any disease you can name, including glioblastoma, the more energy you can put on front end of the problem, the more likely you're going to succeed in curing or at least prolonging life for that disease. Integrated health portals will help monitor things in the background. As hard as this may seem to believe now, industries like the jet engine industry already does this now. You will monitor things in the background and know when a false positive is becoming a true positive. That will then alert an intervention. And of course, lifelong surveillance, learning, and adaptation, including in our schools, to teach health education, will formally transform our view of precision health. This is not precision medicine. This is precision health. How do you keep people healthy for the longest period of time instead of focusing all your energy around treating people? We have approached this through several ways. We've now developed an approach where you will take a pill to determine if you have an early aggressive cancer hiding somewhere in your body. This is called the early detection pill. As weird as this seems, we have proven the basics of this out in animal models, and now a company is taking this forward in humans trials. We inject into you mini circles. These mini circles are a lot like what we've been talking about. They have a switch driving a reporter. The switch only comes on in tumor cells, but when it comes on in a tumor cell, it produces a green protein that ends up in your blood and in your urine. And most recently, we've made this work with a molecule that you'll exhale out in your breath. This specific reporter approach can also be used to identify where cells are, because the same reporter can be imageable, but what you're seeing is a movie in which the reporter is a protein that's detectable in your blood or urine. That lets us know that somewhere in your body there's an early aggressive cancer, and the switch is selected to only come on in cancer cells. In this work, it's the survivin promoter, which is upregulated in many cancer types. We've also recently, after 5 years, developed a way in which immune cells patrolling your body will generate signals when they detect early disease, including cancer. Why not have your own cells constantly patrolling your body and have those cells, in fact, be able to detect when you have detected a given tumor? In this case, we, in fact, remove specific macrophages from you. We modify those macrophages just like you would in an indium-oxime study, but in this case, we insert a reporter that is activated if and only if the macrophage encounters or senses a tumor environment. When it senses that environment, it will activate. So this is different from the other approach in which the sensor is inside cancer cells. In this case, the sensor is inside the macrophages. And the sensor, with the red switch shown there, comes on and leads to, in this case, another protein. And that protein then ends up being detected in the blood. This took us a long time to get to work. A great MSTP student, Amin Anupur in the lab, has been able to make this work. And the secret is in that switch. What switch is specific enough to sense the environment these cells are in? You'll also start to see from us multiple home-based devices. Sungmin Park, a terrific engineer from Korea and UC Berkeley, has been in my lab for 5 years now, as we've started to deploy new nanosensors in toilets that will start to sense molecules being released in your urine that are present and let us identify early disease. We believe these kinds of in-home devices that don't force you to change your behavior, where you can go about your normal business every day, will be the secret to how we will be able to detect disease early through approaches like I'm showing you. So in conclusion, it has been a phenomenal journey since 1996, when I was working actually on decision modeling to help get FDG approved as a normal clinical tool. Through the confidence Mike Phelps had in me and other mentors had in me, we would start to move into this whole area of merging cell molecular biology with biomedical imaging. That would lead to an explosion of applications in multiple fields. And now, as I show you where we are in 2018, we are in a whole era where we're shifting medicine from being more proactive instead of reactive. How do we get to the front end of the problem? And it's been an incredible journey. I've been at the Society of Nuclear Medicine since the age of 20. I'm now 55, and this 35 years to me has been a terrific journey. It's been an unplanned journey. I wish I could tell you we knew where we were going. Each year, things lead to the next thing, which leads to the next thing. But the common theme is that we're always venturing into areas that we're not comfortable with, something I learned to do from people like Mike. Things that make me uncomfortable tell me I'm on the right track. You can learn anything as you need to go. I trained as a mathematician and an MD, not as a cell molecular biologist. But I believe now I can tackle almost any area. Late-stage disease should not be our only focus. We have got to stop focusing on disease at the tail end of the problem. Don't just look at cancer scan after cancer scan, counting the lesions. Think about what we should be doing on the front end of the problem. How do we prevent, and if not prevent, detect very early disease when we have the best chance of making a difference? I believe to do this, a revolution in precision health is coming. And I believe life is much like our joint journey on a train. We're all put on that train. Our parents are on that train with us, but they leave us. In the case of myself and Aruna, our son left us on the train. But it's the journey that's important. And how we treat each other, the respect we give each other, not only to our direct immediate family, but to all of us, to each other, is how we will be remembered. And that's the most important thing we can do in our life as we look after each other as part of our extended family. It's a great privilege to be here to share this stage with the prior recipients of this prize, I had the great honor of working with almost all of them. In yellow hair are the ones that I actually learned from. Henry Wong was, of course, my direct PhD mentor, who gave me a very solid training in mathematics and biomathematics. And Bill Strauss was, of course, right before me at Stanford as the head of nuclear medicine. You know all these people. Mike was a phenomenal inspiration to me and believed in me from a very early age and gave me the confidence to tackle anything I wanted to tackle. All of them are terrific individuals, and I've been blessed and fortunate to learn from all of them. Special thanks to all my colleagues at UCLA, Harvey Hirschman, great mentor in cell molecular biology, who helped me basically retrain in cell molecular biology, Mike, who's been an inspiration from day one, Henry for the phenomenal mathematical training, Dr. Barrio for the phenomenal training in chemistry and radiochemistry, and Sadi and so many others. Thank you to all of them. Special thanks to colleagues all over the world who worked on reporter genes. Special shout out to Ron Blasberg and Yuri Gelovani, who were at Memorial Sloan Kettering, also working on reporter genes, especially with imaging agents like FIAU. And also Mian Aludin, working at that time at USC along with Peter Conti to develop fluorinated acycloguanosines, and many others that have been very important, and I'm sorry that I haven't been able to list all of them. Special thanks to my trainees that were involved in some of this early work and for the great colleague friendship from Simon. Special thanks to our cancer patients and healthy volunteers, to the funding from all our agencies, especially our colleagues also at the City of Hope, Mike Jensen in particular, who's now in Seattle, helping us with a lot of the CAR T-cell work. Andre Agaru, leading now nuclear medicine at Stanford. I like to give autonomy to things that I help build. Andre now leads fully nuclear medicine at Stanford and is continuing to build a great team. Dr. Fred Chin and the cyclotron radiochemistry group, building a great team. And then the molecular imaging program at Stanford, which is about 150 trainees that are from all over the world, helping to build many of the things that we haven't even talked about today. Finally, it's an honor and privilege to lead the Department of Radiology at Stanford University. We're now 800 scientists, physicians, staff that are involved in all areas of radiology, and it's a great honor to continue to lead this department into the next generation. And finally, special thanks to my wife, Aruna, and our dog, Milo, without whom we could not have survived this last few years. Milo was named by our son. It's short for Milagro, which means miracle. And although no miracle occurred for Milan in his fight against glioblastoma, we honor his memory by moving forward in the best way that we can. Thank you all very much.
Now I would like to conclude this session, and please enjoy your meeting. Now exhibitor hall is open. Thank you.